Deep inside almost every cell in your body, there is a structure of unimaginable complexity and beauty. It is a twisted ladder, a spiral staircase made of molecules that defines who you are, from the color of your eyes to the beating of your heart. For decades, we have stared at this structure with awe, able to read it, but never truly able to touch it. But that has changed. We stand on the precipice of a new era in human history. We have found a way to rewrite the code of life itself. This is the story of CRISPR and the future of gene editing. It is a journey through biology, technology, and the deepest ethical questions of our time. To understand the power of this new technology, we must first understand what it is editing. We need to talk about DNA. Think of DNA not just as a molecule, but as the instruction manual for life. It is a massive biological script that determines every physical trait you possess, your height, your eye color, and even your susceptibility to certain diseases. Within this massive manual, there are specific chapters we call genes. Usually, the book is written correctly, but sometimes nature makes a typo. These typos are called mutations. A single wrong letter in a book of billions can be the difference between a healthy life and a devastating genetic disorder. For as long as we have known about genetics, scientists have dreamed of fixing these typos. This is the core concept of gene editing. It is the ability to fundamentally change an organism's DNA. The goal is simple in theory, but incredibly difficult in practice. We want to delete the bad genes that cause suffering, or add good genes to improve health. We want to fix broken genes. For years, this was the holy grail of biology, something that was technically possible, but notoriously hard, inaccurate, and incredibly expensive. But recently, a new technology has revolutionized everything. The revolution has a name: CRISPR. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. A mouthful of a name for a tool that is elegantly simple. To understand CRISPR, don't think of a laboratory. Think of your computer. Think of it as a cheap, easy, and precise word processor for DNA. Just as you can use the find and replace function in a document to correct a spelling error, CRISPR allows scientists to find and replace genetic code inside a living cell. It has democratized genetic engineering, making a capability that was once reserved for billion-dollar labs accessible to scientists worldwide. Where did this incredible technology come from? Did we invent it in a high-tech Silicon Valley lab? Surprisingly, no. Scientists didn't invent CRISPR from scratch. They discovered it. It comes from nature itself. It is, in fact, a natural immune system found in bacteria. For billions of years, bacteria have been fighting a microscopic war against viruses known as bacteriophages. These viruses inject their DNA into bacteria to hijack them. To survive this billion-year-old war, bacteria evolved a weapon. That weapon is CRISPR. Here is how this microscopic immune system works. When a virus attacks a bacterium and the bacterium manages to survive, it does something remarkable. It cuts off a small piece of the attacking virus's DNA as evidence. It then takes this piece and files it away in its own genetic code, saving it in a DNA library. This library is the CRISPR archive. Essentially, the bacteria is creating a criminal mugshot database of every virus it has ever defeated. If that virus ever attacks again, the bacteria checks its files, recognizes the intruder, and prepares to fight. But a database is useless without an enforcer. Enter the machine, the protein known as Cas9. If CRISPR is the archive, Cas9 is the weapon. It is a protein that acts as a pair of molecular scissors. Its only job is to cut DNA with extreme precision. But Cas9 is blind. It has the power to cut, but it doesn't know what to cut. It cannot work alone. It requires a guidance system to tell it exactly where the enemy is hiding. Without instructions, it is a dormant weapon waiting to be armed. This is where the genius of the system lies. The bacteria creates a molecule called guide RNA, or gRNA. This guide is created from those stored mugshots in the library. It acts exactly like a GPS system for the Cas9 protein. The guide RNA holds hands with the Cas9 protein, leading the blind scissors through the complex jungle of the cell. It guides the scissors to the exact location of the matching DNA sequence in the invading virus. It ensures that the bacteria only attacks the invader and not itself. The process is a relentless search and destroy mission. The Cas9 and guide RNA float through the cell, scanning every piece of DNA they encounter. They are looking for a match, searching for DNA that perfectly matches the guide sequence held by the RNA. When they find that match, they latch on to the DNA. This recognition triggers the cutting mechanism. Snap. Cas9 cuts the DNA strand in two. By severing the genetic code of the virus, the threat is neutralized permanently. The virus is destroyed. This bacterial defense system was a curiosity for microbiologists until two brilliant scientists, Jennifer Doudna and Emmanuel Charpentier, realized its true potential. They had a breakthrough idea. If bacteria use RNA to guide the scissors to a virus, could we change the guide? They discovered that by changing the guide RNA in the lab, they could program the scissors to cut any DNA sequence they wanted. We could program these molecular scissors for any species, plants, animals, or humans. This discovery was so profound that it won them the Nobel Prize in Chemistry in 2020. So we can cut DNA, but how does that allow us to edit it? The magic happens in the repair. When DNA is cut, the cell panics and tries to fix it. Scientists can exploit this in two ways. Method 1 is to disrupt the gene. If we let the cell simply glue the cut ends back together, it often does so sloppily, breaking the gene and turning it off. Method 2 is the true edit. Repair with a template. If scientists provide a correct piece of DNA alongside the CRISPR tool, the cell will use that good template to patch the hole. This allows us to fix a mutation by physically pasting in the correct code. This is not just theory anymore. We are seeing the first real-world application, curing disease. Take sickle cell anemia, a brutal genetic disease caused by a single-letter typo in the DNA. In 2023, the FDA approved a therapy called Casgevy, marking the first functional cure using CRISPR. Doctors remove a patient's stem cells, use CRISPR to edit them in the lab, fixing the genetic issues or reactivating healthy fetal hemoglobin, and then return the corrected cells to the patient. It is a life-changing intervention for people who previously had no hope. The revolution extends beyond medicine and into agriculture. As our climate changes, we need crops that can survive. Scientists are using CRISPR to create crops resistant to drought and pests, ensuring food security for a growing population. They are also making food last longer, creating mushrooms that don't brown and tomatoes that stay fresh. Importantly, this is different from traditional GMOs. We aren't dragging in foreign DNA from fish or bacteria. We are simply editing the plant's own code to make it stronger. Then there are the applications that sound like science fiction, de-extinction. Scientists are working on bringing back extinct animals. By taking the DNA of an Asian elephant and editing it to resemble its ancient ancestor, the woolly mammoth, they hope to create cold-resistant elephants. The goal isn't just a Jurassic Park-style spectacle. It is to restore Arctic ecosystems. These massive grazers could knock down trees and expose the ground to cold air, helping to keep the permafrost frozen and fighting climate change. Why has CRISPR taken over the world so quickly? It comes down to the power of precision. Old methods of gene editing were like using a sledgehammer to crack a nut. They were imprecise, incredibly expensive, and required advanced facilities. CRISPR is different. It is like using tweezers. It is fast, cheap and accessible. Today, even high school labs can use it. This democratization means innovation is happening everywhere, but it also means the Pandora's box is open to everyone. This accessibility brings us to a massive ethical dilemma: germline editing. We must distinguish between two types of editing. First, there is somatic editing. This involves editing adult cells, like the liver or blood. These changes affect only the patient and die with the person. They are not passed to children. This is generally seen as safe and ethical. But then there is germline editing. This involves editing embryos, sperm, or eggs. If you make a change here, it changes the human species forever. That edit is passed down to children, grandchildren, and every generation to come. Germline editing opens the door to the concept of designer babies. If we can fix diseases, can we fix other things? Could we edit for height, eye color, intelligence, or muscle mass? The line between therapy, fixing a disease, and enhancement, improving a human, is blurry. This raises terrifying questions about equity. Will we create a gap between the genetic rich and poor? Will only the wealthy be able to afford perfect children, creating a biological caste system? These are questions we are not yet ready to answer. Aside from ethics, there are real safety risks. We call these off-target effects. CRISPR is accurate, but it is not perfect. Sometimes the GPS gets confused. It might find a DNA sequence that looks similar to the target and cut the wrong gene location. These accidental cuts are dangerous. If CRISPR snips a tumor suppressor gene, it could accidentally cause cancer. This safety risk remains a major hurdle that scientists must overcome before widespread human use becomes a reality. As the science races forward, the laws are struggling to keep up. We face a problem of regulation and the future. Currently, there is no global consensus. Different countries have different laws regarding human embryo editing. What is illegal in one nation might be permitted in another. The technology moves faster than the laws. While the global scientific community largely agrees that we should not edit embryos for pregnancy yet, the potential for rogue actors remains a constant concern. We have come a long way from a simple bacterial immune system. In summary, CRISPR is a tool borrowed from bacteria that uses molecular scissors and a GPS guide to rewrite the code of life. It holds revolutionary potential to cure disease and feed the world, but it comes with heavy ethical questions. We now hold the pen that writes the story of life. The question is no longer just about what we can do. The question that will define the next century is, just because we can edit humans, should we?